Welcome back, future healthcare professionals. So, in your last lesson, you tackled the biological characteristics of microbes and, their massive environmental impacts. But right before we dive into the dark side of microbiology to look at infectious diseases, which is coming up next, we really need to bridge that gap. In today's explainer, we're going to flip the script and look at our greatest clinical allies. We'll see how the exact same types of organisms that cause disease are also, incredibly, the absolute backbone of modern medicine and industry. Okay, so how do we harness this immense power before we start studying the actual pathogens? I want you to completely shift your perspective. Step away from the natural environment for a second, and instead picture a massive, super efficient factory floor. We're going to explore how we take these microscopic organisms and use them as highly programmable biofactories. Here's our roadmap for today's explainer. Number 1, the Microbial Biofactory. Number 2, Bioremediation and industrial scale. Number 3, manufacturing pharmaceuticals. Number 4, genetic engineering, the blueprint. And finally, number 5, clinical diagnostics and immunology. All right, let's jump right into section 1, the Microbial Biofactory, scaling up nature's machinery. So at its core, biotechnology isn't really about reinventing the wheel. It's more about hijacking it. We take a living organism's totally natural metabolic pathways, and we just deploy them on a massive scale. We're basically taking their inherent biochemical processes and scaling them up to create the food we eat, the drugs that cure us, and absolutely crucially, the vaccines that protect us. Moving on to section 2, bioremediation and industrial scale, the heavy machinery of microbiology. You really have to appreciate the sheer scale of these operations. On the one hand, we've got industrial microbiology scale. This is where we precisely scale up microbial reproduction to synthesize just massive, commercially viable quantities of specific chemicals or biological products. It's all about building things up. But then, on the other hand, look at bioremediation frameworks. This relies on catabolic processes, tearing things down. We systematically introduce highly specialized microbes to degrade, neutralize, and literally chew through severe environmental toxic waste. So we're talking about two vastly different applications, building up and tearing down, but both are powered by the exact same microscopic machinery. Okay, on to section 3, manufacturing life-saving pharmaceuticals. This is our medical assembly line. In the medical field, we can divide this microbial power into 3 really critical applications. First up is pharmaceutical mass production. We use microbes to turn out critical everyday medications like human insulin, growth hormones, and anti-inflammatory agents. Second, we have antibiotic biosynthesis. Now, out in the wild, microbes compete fiercely for resources, right? Well, we take advantage of that evolutionary warfare. We cultivate specific soil-dwelling fungi and bacteria that naturally produce compounds designed to destroy the cell walls of competing pathogenic bacteria. It's a brilliant strategy. And third, vaccine manufacturing platforms. This is where we engineer benign viral vectors or expression systems to cultivate targeted surface antigens for our immunization programs. Let's look at how we actually do this in section 4, genetic engineering, the blueprint, reprogramming the factory. Before we get into the nitty-gritty molecular mechanics, we really need to understand the end goal here. We are deliberately moving an organism away from its native DNA sequence, its original genetic code. Through precise molecular manipulation, we transition that organism into a totally new state to create novel microbial behaviors. We're structurally modifying them to have completely unique physiological traits, tailored specifically for things like therapeutic delivery or diagnostic tracking. And a huge part of this is recombinant protein synthesis. This is wild. We are literally taking human therapeutic genes and dropping them directly into bacterial or yeast expression hosts. By doing this, we force these microbes to act as cellular factories that read our human instructions and translate those genetic blueprints into functional, life-saving human proteins. Let's break this down with a classic car factory analogy because it makes the process super clear. Step 1, you have the standard blueprint. The bacterium's native circular DNA is like the factory's standard instruction manual for building, say, basic everyday sedans. Step 2 is the snipping. We cleanly cut out a page from a human manual. Let's pretend it's the exact blueprint for a premium stereo system, which in our case is human insulin. Step 3 is pasting. We insert that premium stereo blueprint directly into the bacterial instruction book. And then step 4 is mass production. The factory workers, the bacterial ribosomes, they don't care where the instructions came from. They're totally blind to the origin. They just read the new blueprint line by line and start cranking out mass quantities of premium stereos right alongside their regular sedans. It's just a brilliantly efficient system. Finally, let's look at Section 5, Clinical Diagnostics and Immunology. Our body's security system. We don't just use microbes to make drugs, we actually use their very components to build the clinical assays that keep our patients safe. Looking at immunological monitoring, we harness the host immune system's complex web of substances to identify active systemic exposures to pathogens. For blood testing diagnostics, we utilize purified microbial antigens in clinical assays to detect specific circulating antibodies in a patient's serum. Basically, we use pieces of the microbe to figure out if the patient has been fighting that exact microbe. And lastly, for allergy and vaccine formulations, we isolate and deliberately modify microbial proteins to formulate highly accurate allergen panels and those incredibly important protective vaccines. So, as we wrap up this explainer, I really want you to lock this in. We have completely mastered using microbes as programmable biofactories. We use them to scale up industrial production, clean up our environment, mass-produce human insulin, and build complex diagnostic tools. They are, for sure, our greatest clinical allies. But I want you to keep all this incredible bioengineering power in the back of your mind as you step into your next lesson, because you're about to dive into pathogenic microbiology and the burden of infectious disease. You're going to see exactly what happens when this very same machinery gets turned against us, and the pathogens fight back. Good luck, and I'll see you in the next one.